talk of microscope and microscopy. A very tricky area indeed. You realize that we normally start microscope work in Form 1, but by the time we get to Form 4, maybe because of constraint of time, hardly do teachers have enough time to polish on microscope. So what I want us to do today is to go through some things that people may take for granted, and you realize that they're very, very vital. Some of the aspects that we are handling here may also help us even in theory work, but mainly are concentrate on the practical aspect. But you realize that you cannot thrive in a practical unless you have the backdrop. That is the theory. So I will not take much time dealing with the parts of a microscope, but we may, in a very quick way, go through the microscope, and especially the light microscope. Of course, we normally use microscope to observe cells, which are too small to be seen with naked eye. That's what we say. Now, at the same time, because this is quite key, objects that are too small to be seen by an aided eye. Therefore, our microscope that we use is what we call light microscope. Now, most students have a challenge with operating a microscope, and now that we don't have one here, what I would like to encourage you, because an opportunity will present itself. You cannot operate a light microscope unless, number one, light is passing through the lens, through the diaphragm, then through the condenser, then through the stage, then through the objective lens and the eyepiece lens. Unless the microscope is set in a way that light can enter. I saw agony in students in 2014, yeah, when students were given a microscope in KCSE, and you would see students struggling. They would see darkness. Maybe that is something that we want to emphasize, that anytime you are given an opportunity to have a microscope, to use a microscope, the first thing is lighting. Make sure that it is well lit. And how do you ensure that our microscope is well lit? Number one, you make sure that the eyepiece lens and the objective lens have clicked in position. You know, that's the key, that both eyepiece lens and objective lens have clicked in position. Number two, you make sure that the diaphragm is open. Most students struggle with a microscope whose diaphragm is closed. And you see, diaphragm is the one that regulates the amount of light entering the stage. So in case you start with a microscope whose, number one, the lenses are not aligned, especially the objective lens has not clicked in place. Number two, the diaphragm is closed. Number three, the mirror is not reflecting anything. The mirror is not reflecting anything. Those are the key, very key areas that most students go wrong. And if you pay attention to those main areas, you always have an easy time. But anyway, as we move on, that is something that we can always work on. So maybe just a crash labeling of our microscope. One is eyepiece, as I'm doing that. Number two is coarse adjustment knob. Then we have fine adjustment knob. Then we have the limb or the arm. That part is called limb or arm. I know you know that. Then we have the condenser. Then the diaphragm, and the spellings must be accurate. Then we have the turret or body tube. Turret is also known as body tube. Then we have the revolving nose piece. Yeah. Then, of course, those are the objective lenses. That is the clip, that is the stage, and of course, that is the mirror. Now, I don't want to go through the parts, but this is one area I want to challenge you. You know, the candidate of 2020, in as much as there is a crisis, I would like us to look at the crisis as an opportunity. If you look at now, the source said the 6 more months added to us to be at home. If you take those 6 months as a challenge, as a problem, I tell you, you may not translate it into something. Um, you may not actually convert that time into something constructive. I would like us to look at the remaining time as an opportunity. And that's why, if you look at these parts, they're quite many. And of course, the wordings, the words that you use, the choice of words that you use with regards to the role of each of these parts. We may not do it here, but I want us to challenge ourselves and use that time, which looked like it is too much, with an aspect of an opportunity. Now, where need be, we may mention one or two. Now, functions of a microscope. Now, there are two main functions of a microscope. Number one is magnification, and number two is resolution. Now, since I want to take maximum time dealing with the practical aspects, but I'll keep repeating and saying even the theory is very crucial. But it's also good to know how even we tell the difference between these two. Magnification, yeah, process of making very small organisms appear bigger, because you realize that even in practical, you may be asked some theoretical question, as we have seen in the past. I'm going to show you as we move on. Now, making very small organisms appear bigger. Most students would say that magnification is making small organisms bigger without putting emphasis on the word appear. If you don't emphasize on the word appear, of course, that now will translate that a microscope can make an insect look like a member of the other phyla, members of phyla, chordata. An insect cannot be made big by a microscope. What microscope does is to make it appear. That's the word, to appear bigger. Even the word enlarge is wrong. It is basically to appear bigger, because in real sense, it is not enlarging. And I hope that is well taken. And as we move on, we go to the solution. Now, again, just to test my speakers and my microphone, my speakers at least, maybe I want to ask somebody to tell us what the solution is. Somebody have not asked a question for some time. Amanda Ngujiri, what is the solution?
I'm not sure.
Yes, tell us what you are not sure, Amanda. Just tell us what you have in mind.
I think it is the appearance of the organism after it is already, I don't.
Amanda,
I'm not sure.
you're trying, but that's okay. Thank you for trying, Amanda. Let me try the last one. Wale ninajua ama wale sijui. Quincy, kuna mwanafunzi anaitwa Quincy. Yes.
Resolution is the process by which a microscope distinguishes two items which are very close as distinct entities.
Quincy, I think Quincy has tried to make it more clear. She has resolved. And what Quincy has done is to resolve, to make it clear. That's good. Thank you, Quincy. Hata Amanda amejaribu. Now, resolution is separating objects that are close to one another. By this, I mean, separating objects that are close to one another, or simply ability to give fine details in a structure. Ability to give fine details in a structure. Now, I want to illustrate that practically. With us here, we have an animal cell under light microscope. That one, an animal cell under light microscope. But if we use an electron microscope, which has higher resolving power, you realize that we move from seeing only 3 organelles. Here we are seeing plasma membrane, nucleus, cytoplasm, quisha, nothing more. But when we resolve using an electron microscope, you realize that we are seeing more details, organelles that could have otherwise been not seen. Here we can even see the Golgi apparatus, we can see the nucleolus, we can see the nucleopole. You know, like now the nuclear membrane here is not it's not even visible. Where's the honor? But when we resolve, we can see that even nuclear membrane is double membrane. We can even see that nuclear membrane has pores, what you call nucleopoles. We can even see that inside there, the nucleus, we have nucleolus, even what you call nucleoplasm. We can see mitochondria. We can see lysosomes. We can see Golgi apparatus. We can see ribosomes. You know, that is now resolution. Moving from something that was not very detailed to something detailed. And I hope you can see now where the word details come. Not just detail, fine details. Ability to give fine details in a structure. So, 2 roles of a microscope, magnification and resolution. And as we move on, we are going to see this more and more and see how we can apply some of these techniques and terminologies. I hope that is now very clear. So I've gone ahead to and illustrate it even more as we go on. So this to make it possible to see what 2 objects which are very close to one another, a separate object. We call that a resolution. So we talk of resolving power of a microscope. Now, I know, you know, microscope being the icon. This is the icon of biologist. If you visit somebody's office and you find a microscope, you know that, oh, this is a biologist. So this is our tool of trade and we should understand it in depth. So parts of a microscope, that's what I'm saying. I may not go through details right now. This is more theory and it may take a lot of our time. So limb, arm, to support the body tube. Next, base, the lower heavy part of the microscope. It rests on the bench and give microscope firm support. I want to encourage everybody, please, take these nitty-gritties into context of your revision. It will empower you more. Eyepiece, or eyepiece lens and or ocular lens. And now, when you talk of eyepiece lens or ocular lens, it magnifies further. Look at that. The reason why I've highlighted it in red, most students don't take those details into account. It magnifies further the image formed by the objective lens. We know that microscopes start working from the lower part, the objective lens, then going upwards to the eyepiece lens, this one. But remember, we have eyepiece and we have eyepiece lens. The whole of this structure now is the eyepiece. Yeah, this part is the eyepiece where I'm putting the spotlight. Then in the eyepiece, now we can insert the lens. So this lens can fit in here. So always when you're communicating, communicate precisely. Are you talking about the eyepiece or the eyepiece lens? So most students don't distinguish that. So eyepiece now holds the eyepiece lens. That's something that we need to know. Now, remember that the eyepiece lens can be removed and replaced with another eyepiece lens. In fact, the eyepiece lens can be interchanged. They're interchangeable, meaning when anytime you're handling a microscope, you know that if you hold a microscope upside down, the eyepiece lens will fall. Itaanguka, yeah, it will fall. And that's why as we handle a microscope, you do it with a lot of care. And therefore, it is always good to be conscious of which eyepiece lens are you using, which power, because it's something that can be interchanged. And getting to that, of course, the most common eyepiece lens, they are either times 10 or times 15. So when using a microscope, take into account which eyepiece lens are you using. Without that now, you'd have missed a very important point. Now, then the objective lens, and we have them there. Now, when you talk of objective lens, kuna watu wanashindwa kuingia. That's why I think it's good to enter early. Because I think I hope there was no challenge, unless somebody has a network problem. I think the majority of us have managed with very easy. It's not like last time. There's a time when there was a challenge in the network. I hope today it's not the same. Let the person keep to try a bit for some time. Now, when you talk of objective lens, these ones now bring image of the specimen into focus and magnifies it. If you look at now the functions of these two lenses, they are different. Objective lens bring image of specimen because it's the one which is just next to the specimen and magnifies it. But if you go back to the role of eyepiece lens, it magnifies further. So many people miss that point, that aspect of further. So it magnifies further the image formed by objective lens. So you can see the role of eyepiece lens is making reference to objective lens. Now, those are the nitty-gritties that I want us to, also, of course, focus in, so that they make us superior students. Eyepiece lens make reference to objective lens, but for objective lens, it brings the image of the specimen into focus and magnifies it. So please, even as you go to your theory, make sure that you capture that very clearly. Hiyo ni homework. Now, light microscopes, the one that we use in school, have three objective lenses mainly: the lower power, the medium power, and the high power. Now the low power, as you can see on the photograph, it mainly has a magnification of ×4. Now, kuna kitu sasa ningependa tuchukulie very seriously now at this juncture, because it has a lot of practical implication. Now, when you're using the lower objective lens, you are going to observe the largest area of the specimen, what we call the largest field of view. Na hii ni kitu sijui, I know I've done it down there, but anytime you are using ×4, with whichever, with either ×10 or ×15 up there, this is the time you're going to see the largest area of the specimen, or the widest field of view, if I may put it that way. Because we normally have a rule, and this rule, I normally use my hand this way. I say the lower, this is the way, yeah, the lower the magnification, the wider the field of view. Look at what I'm doing with my hands. The lower the magnification, the lower the field of view, the wider the field of view. I think instructions, I think we had communicated that we are starting at what time? At 7. Na mtu alianza kulogin 6:30 na amekuwa frustrated. Na tumesema vizuri. I think it's for confusion. So allow me to so to agree kutoka last time we start at 7. Somebody started logging in from 6:30. Amekuwa na shida huyo kwa sababu amesafa sana. I think that's where the confusion is. Hello, you allow me to make. Yeah, technology inakuwa na vitu na mambo. So I was saying the lower the magnification, the wider the field of view. Yeah, the more you go down, and you can see now my hands are opening up, the wider the field of view. And by that I mean you see more cells and you don't see details. So that's now the other aspect. You see a lot or even larger part of the specimen, you see more cells, but for details they become less and less. The resolution become less and less. And the converse is true. The converse is true. The higher the magnification, the lower the field of view. So inaenda ikiwa kidogo. So you count more cells. No, you count fewer cells, but you see more details. So the converse is true. The higher the magnification, the lower the field of view, but the more the details, the higher the resolution. So make sure that you get that idea. If you miss that, it will not be so smooth for you. And now, medium power, mainly it magnifies times 10. Of course, with the product of the eyepiece lens, we'll talk about that. And we always resort to medium power after. And here, the protocol is very important. Anytime you start observing a specimen with a microscope, always start with the lower power. Always start with the lower objective of times 4. And you see here times 4, interpret times 40 because up there is an eyepiece lens. If you don't start from lower power to medium power, you'll be frustrated. You may not even focus or land on the specimen that you are looking for. And that's why here we are saying it is used after the low power lens has been clearly focused. So if you breach that protocol, you'll be frustrated. After we clearly focus the low power, then we go to the medium power. And as usual, because of the rule that we have tried to engage here, it shows smaller area of the specimen, but with more details. When you talk of smaller area, let's say the lower power had given us about 16 cells, let's say across the diameter. This one may give us only 5 cells, from 16 cells to only 5 cells, but with more details. That means with higher resolution. Then we go to high power. Now high power of times 40. Here there is now a challenge a little bit, although it is still useful in our context. But sometimes when you use the high power now, unless you engage another technique known as oil emulsion, and in high school, maybe unless you're given the details to use oil emulsion. Because we normally, after we mount our specimen, which we will mention, we always add a drop of oil we call it oil emulsion, so that it can concentrate light more. And unless you use oil emulsion, we may not see a lot of details. You may not see the specimen clearly. But I remember in 2014, students were instructed to use high power, even without oil emulsion. You will see something, but to enhance whatever you observe, your image, we normally add oil, what you call oil emulsion. But it's a technique that we don't mention nowadays in high school. But who knows? In one of these exams, an examiner may introduce that. But in 2014, students used the high power without oil emulsion. So here we see very small area of specimen, but with most details. So this is now where you can see structures that we always talk about in theory. And we did that in 2014, because in 2014, we were observing the yeast cell. And we shall see the question of 2014 in a short while. Now, with that in mind, we have other parts, which I don't want to spend more time on that. But this part, which you call the revolving nose piece, sometimes called the turret, it's also very important, because unless you operate the turret effectively, that is moving from one objective lens to the other. One of the biggest challenge is that as you move from low power to medium power, and you rotate this part, most students give up very early. Yeah, they give up very easily. Remember this part, when you are switching from one objective lens to another, unless it click into position, and you must even hear that sound, the clicking sound, you may end up focusing on the wrong thing. Our lenses may not be aligned correctly. And there was a question in 2015, and it's in one of our slides down there, where the examiner in 2015 was so much interested on how students operate this part, which we are calling the turret, the revolving nose piece. So when we get there, I'll spend more time mentioning what the examiner wanted. And anytime you see examiner bringing a question of that nature, is because out of experience, we have seen students having a lot of challenge in that area. Then, of course, the stage, the flat platform onto where the microscope slide with the specimen is placed, and of course those clips. And that stage must always be be dry. And I'll take note of that point I've just said. That stage must be dry, because if it is not dry, you experience a lot of problems with regard to diffraction of light. Utawekelea slide and it's going to refract light. You may not see the specimen clearly. Then the body tube. Of course, the body tube, the one that will move up and down when we are rotating or when you're operating on the coarse adjustment knob and fine adjustment knob. Now, when you're operating the knobs, when you're operating the knobs, the body tube move up and down by either coarse or fine adjustment knob. Make sure that you are observing also the stage and the slide, because you can easily break the slide, especially when you're using the high power. It's very easy to crash your specimen. So as you do so, make sure that you are observing everything clearly. And maybe on that note, coarse adjustment knob, anytime that you are observing a specimen, don't start operating the fine adjustment knob. You always start with the coarse adjustment knob. Reason, it's the one that will now move the body tube. The body tube up and down. We normally say into larger distances. Into larger distances. Bringing the specimen into focus. Nowadays, even the books are saying bringing the specimen into rough focus, but we say it into focus. Now, once you focus with the coarse adjustment knob and you start seeing something that is real or that is making sense, that's when now you resort to the the fine adjustment knob. So if you start with the fine, you will not go anywhere. You always start with the coarse adjustment knob. Once you see there is a rough idea of what you want to see, then now you resort to the fine adjustment knob. You know, those manipulation skills, they may look very simple in theory, but in practice, they are a nightmare. They are a nightmare. So that's another area that you cannot afford to ignore. Yeah. And even knowing how to articulate the role of this part, because it is after you know how to articulate the role, now you can put it into practice. So it moves the body tube into larger distance, up and down into larger distance, bringing the specimen into focus. Now, down here now we go to fine adjustment knob. And here the fine adjustment knob, depending on the microscope, it could be this part here. The coarse adjustment knob is the bigger one, then the smaller one inside here. Now this is the fine adjustment knob. Of course, this one moves the body tube up and down into smaller distance, bringing the specimen into sharper. Okay, here it is not well put or said. It should be sharper focus, bringing the specimen into sharper focus. And that's now when you now realize exactly what you wanted to observe. So to bring the body tube up and down into smaller distance, bringing specimen into sharper focus. And after that, now you stop and you start observing. If the examiner tells you to draw, you draw. Now, there is a lot that, of course, the mirror, we said the mirror must be well placed so that it reflects light into the condenser, then into the diaphragm, into the condenser, then into the diaphragm, then into the condenser, so that it can light into the stage. And as simple as it can be, it can frustrate you if you don't adjust it accordingly. Now, I want to say something about the diaphragm. Now, some microscopes, because diaphragm is the one that regulates the amount of light that enters the specimen. In fact, diaphragm, how does it regulate? Of course, you can see here in what we call the central circular opening area. You can see this part, it's like the parts are overlapping. The moment you operate this lever, this is called a lever. The moment you push this lever up, these overlapping canals, circular areas, they are going to close, or they are going to alter the size of the opening here. They are going to either to increase the size or to decrease the size of the opening. Therefore, effectively regulating the amount of light that enters the specimen. And that's why some literature may call it iris diaphragm, just like the iris of the eye. Hapo nimekuchorea jicho hapo, the structure of the eye. Just like the iris of the eye, which regulates the amount of light that enter into the retina. Ukiingia kwa dark room, your iris dilate, okay, your pupil dilate because the iris will have muscles that regulate the same. If you go to a room with very bright light, the muscles of the iris will contract. Okay, there are two sets of muscles. Let me not go to the details, the circular and the radial muscles. But the general perspective is the iris of your eye regulates the amount of light that enter. Likewise, the diaphragm is like the iris of your eye. That's why some books may call it iris diaphragm. And most students forget, anytime they get a microscope, they forget to operate the iris, the diaphragm. If you don't know how to operate it, you better even ask for help. Because if you may operate with a microscope that has a diaphragm that is closed, imefungwa, na unaona giza and you don't know why. Some microscopes, the diaphragm is down there below the stage. Some microscopes, the iris is, the diaphragm is just next to the stage. It look like a knob ya TV ama ya radio. Ukizungusha unaona mwangaza ukiwa mingi ama ikipungua. So make sure that anytime you have an opportunity to operate a microscope, you see those parts very clearly. In fact, as I've been promising, when we go to another level in this forum, we will even use this apparatus live here. Then on the other hand, condenser, we hardly see the condenser because it is down, it is inside. So we hardly see it, but this is the one that concentrates the light to the stage. So condenser concentrates light to the stage. Now, in 2015, students were brought this question. And this is a question that is touching on how we manipulate a microscope. It is indeed emphasizing on manipulative skills of a microscope. The question reads, how is the low power objective lens manipulated to focus a specimen for observation under a light microscope? 2 marks. I repeat, how is the low power objective lens manipulated to focus specimen for observation under a light microscope? Now, you see, that one sometimes can be a tall order to somebody who have not been working on a microscope. Now that it's a random question, let's hear. Nastaki mtu asome kwa kitabu. Acha niulize. Lilian Nyaga, what can you say? You want to try? Lilian Nyaga. Now, Johnson.
Yes.
What can you say?
Okay.
Yes, tell us anything. Okay, fine. It's all right. Somebody said it's okay not to be okay. So it's okay because we learn through mistakes, but you have tried. Now, in the interest of time, look at this statement. Click the lower power objective lens into position. Yeah, bring it to the lowest level using coarse adjustment knob. With the eye on the eyepiece lens and using the coarse adjustment knob, gradually raise or lower the power objective lens. Yeah, with the eye, and you can see the details. Even you are supposed to know where to place the eye. With the eye on the eyepiece lens and using the coarse adjustment knob, gradually raise or lower the power objective lens to bring the specimen into focus. And let me give you time to internalize that. Click the lower power objective lens into position. In bracket, that means bring it down to the lowest level using coarse adjustment knob. That's the next. With the eye on the eyepiece lens and using the coarse adjustment knob, gradually rise or lower the power objective lens to bring the specimen into focus. Meaning, we are not talking about bringing it into sharp focus. And that's why we are not talking about the fine adjustment knob, because the question has indeed directed you how far you should go into focus, not into sharper focus. sharper focus to the ceremony when using the fine adjustment knob. So the question is that simple, although it can be tricky. I don't want to underrate the question, it can be tricky. Now, with that in mind, this tells us that hii kazi ya microscope ni mingi. There are so many nitty-gritties that we need to bring our house into order. Hii mambo ya microscope, that one I know. It is an area that even an expert like me can have some challenges here and there. But with a lot of practice, it can become easy. And you can see now, this will happen once. To you as a student, it will happen once. And now, so the more prepared you are, the better, because there will be no room for adjusting after you have messed in an exam. So that's why we should prepare ourselves to the best of our knowledge. And I'm still going to repeat, especially to the candidates of this year, the longer the period that God has given us, we should take it as an opportunity. Let's take this period as an opportunity. Now, I want to also talk about the field of view. I know I explained this, so I don't need to repeat so much. Field of view, the circular space in the microscope in which the image of the specimen is observed. Na nikasema that the higher the magnification, the smaller the field of view, but the more the details. And by that I mean, you can see now, like now at times 150 magnification, we have only 3 millimeter marks. Yeah. So that is to show that the field of view is smaller. But also we should not forget, the more the details we see, the higher the resolution. On the other side, at times 75 magnification, we have, that is lower magnification, of course, but the field of view is wider. We can see about 5 millimeter marks, but also the details are now few. So that is something that I want to emphasize all through. So field of view varies according to magnification at which the specimen is viewed. And we are going to see the implication of this, of such kind of an observation in an exam scenario, as we had in 2001 KCSE. So under low magnification, the power of the field of view is wider. Sorry, the field of view is wider, but that magnification is low, but the details are fewer. The resolution is lower. Now, I don't want to go on and on about that because you to be less vizuri. Now, let's now narrow down to some areas that the examiner may be interested. Before now we come to what we call a microscopy. I'm about to talk about microscopy, the skills that we need to develop so that we can answer questions effectively. One of the questions that one may be asked is, number one, to determine the number of stomata on lower and upper epidermis of a leaf. Now, if an examiner wants you to determine the number of stomata, and a student don't know even how stomata look like, that one now will become a challenge from the word go. But here we have an epidermis of a leaf. I remember I once gave some student this kind of a question, and I found one girl literally taking a leaf and placing it on the stage. Imagine, sijui kama unaelewa venye ninasema. Anachukua hiyo leaf, anaiwekelea kwa stage, anaanza kufunga jicho moja, anakufocus. And you see now, here, now, it actually now took me aback, and I realized how sometimes we, even as experts, we take some things for granted. Of course, when you're given a leaf, the first thing that should come in your mind is how to peel. We peel. Sometimes the examiner may not tell you how to go about it. But now, like this question, the examiner is telling us, how to peel. Peel off the epidermis of the lower surface of the specimen, mount the portion of the epidermis on water, okay, all those details, stain. So I want us to see where those terms come now, where they take us. How do we peel? How do we go about peeling? How do we mount? Now, you see now those terminologies. What type of what type of specimen or what type of slide are we making? Yeah, because there are different type of slides that we need to know. So I want to use this now as our platform to go down to the microscopy, the techniques that we need to know when handling a specimen and then mounting it on a microscope. Of course, for instance, and this is a very common part of a leaf that everybody should know, even how it appears. But it's very easy to make a slide and observe stomata. Sometimes you do so with students and you realize that some are struggling even to identify the stomata. These ones. Meaning this is a guard cell, this is another guard cell, then at the center there is the stomata. Yeah, this is a guard cell, this is a guard cell, then at the center there is a stomata. Is a stoma, sorry, is a stoma. When it is one, is a stoma. Zikiwa mingi ndio tunaita stomata. So I want us to move on, and I know this is an area where most students, in real sense, they feel very uncomfortable when they are dealing with this kind of work, but it's the reality. So that is lower epidermis, and this is what you encounter when you observe upper epidermis. It's also good to be very conscious. Anytime you handle a leaf, no, I'm peeling from lower epidermis, and this is what I'm anticipating. Of course, in most plants, stomata will be found more on lower epidermis than on upper epidermis. Slides from upper epidermis may look like this, because if you look at these cells now, these are epidermal cells with no stomata at all. Because most plants will always strive to conserve water, because in case they have more stomata on the upper epidermis, the more they lose water. So a slide on upper epidermis in most instances, unless you are dealing with a hydrophyte. If it was a hydrophyte, it will be now the vice versa. Because from our ecology, we know that hydrophytes have a problem of having excess water. And that's why now they need to have more stomata on the upper epidermis so that they can lose excess water. Because now, any slide that you make now, the examiner now may go to the idea of even asking questions that border the ecology, the habitat. From what habitat does this plant come from? Then explain. Just because of the number of stomata on the lower or upper epidermis. So that is an area that we need to infuse even as we prepare our slides in future. Now, then another thing, and this is an area that most students again have problems. This one mainly coming from 3, because we can also use microscope slides to demonstrate mitosis and meiosis, or simply cell division. Yeah, or simply cell division. And one of the unique thing about this kind of a slide, when you're making a slide to observe cell division, is even the chemicals that we use. They are not the ordinary chemicals. We have hydrochloric acid, then what you call acetic or rain stain, then Bunsen flame, then spirit lamp. Now, of course, now this borders another technique that we are going to see much later called fixation. Sometimes in Form 1, we ignore aspect like fixation. And anytime you say hydrochloric acid, acetic or rain stain, mainly the examiner may be looking for you to observe aspect of cell division, that is mitosis and meiosis. Although we have never had that one in KCSE, but it is worth noting. And in Form 3, I don't know how many made this kind of slides where you observe the growing region of an onion. You observe onion cells that are actively dividing, which can help us to even understand the stages that we talk about, the metaphase. Now, and you can see now details here where during metaphase, how the chromosomes will align themselves along the equatorial plane of the cell. With the theory, you can easily identify the cells that are dividing. At anaphase, yeah, where now they are now the sister chromatids are pulling to the opposite poles of the cell. Then at telophase, now when the cells are dividing exactly as you can see here. I know in 2014, we saw some aspect of cell division in KCSE, what you call budding. You know, during budding, budding is more or less like what we call cell division. In fact, it is cell division. So I'm talking about this because indeed we have seen it happening in 2014, budding. So don't ignore that practical of observing mitosis and meiosis that we handle in Form 3. Because an examiner can infuse that with now the practical, like the one for respiration. The one of 2014 was respiration. And you see that is Form 2 question. A Form 2 question and a Form 3 question at a go, happening at once, where now you're supposed to handle respiration, it is in use. But at the same time, the question is also about reproduction. So those nitty-gritties are very important to understand. And in Form 3, that is when we normally handle this process of mitosis and meiosis. And indeed, we are supposed to do the practical aspect of that and even make those slides. Then the other aspect that an examiner always brings is to estimate the cell size in a leaf. Estimating the cell size in a leaf. Now, mainly what we normally use is Tradescantia. I'll talk about that one in a short while. In fact, if you allow me to, I wanted to infuse this with something else. Yeah. Now, when you talk about estimating the cell size, what we should remember, and indeed, I want to use a real specimen that I have captured. So I think I better go down. If need be, I'll come back. I think I'll come to that because I have a slide that is down there, and I've done some work about it. So I think you will allow me to do that later because I have better slides than what I have here. So to estimate the cell size, I'll tackle that in a short while. Now, the other aspect. Now, if we are dealing with the KCSE of 1996, there was a very unique dimension where students were supposed to count. Okay, I want to mute this person. No, so student was supposed to kindly mute. I'm trying to mute the person who is talking. So now in 1996, for instance, an examiner wanted us to peel the epidermis on lower and upper side of the leaf. And in this case, it was sukuma wiki. And after you peel and make the slides, we were now to count the number of stomata on both lower and upper epidermis, and now compare which of the two has more. And therefore, an examiner is expecting us even to know how to count. And I want to narrow down to this one, because when it comes now to counting, if you are brought now a slide like this one, yeah, let me start with this one. And you are supposed to count the number of stomata. You see, Unless number one, you know what are stomata, you may have a challenge. Like in this case, I can just point them. This is a stomata. This is another one. The one that I'm doing in green. There are quite a number. This one, this one. You know, most students now will complain, I can't see, I can't see. So if you can identify, then you can count. And the reason why sometimes this kind of a question become a challenge is because you are supposed to move from low power to high power as instructed. If an examiner wants you to count using low power, you should now remember that you are likely to get higher number. You're likely to get higher number of stomata. Reason of what we said. We said the lower the magnification, the wider the field of view. So the implication is that if you use low power, you're likely to see more, to count more specimens. But if the instructions were to use medium power and you use low power, that means you are going to get a totally different figure against the marking scheme. So, and that's why I said those nitty-gritties can be very costly if you don't play by the rules. Examiner akisema utumie low power and you don't obey or you don't stick to that, you get your own figures. And therefore, during marking, you fail. Utaanguka tu, not because it was difficult, but because you never played by the rules. Like now, if we move from this, now this is low power, of course. This is low power. Let's see how it would appear. The same slide would appear if you go to a medium power. If you increase magnification, of course, it will be something like this. Whereas we were getting about 15 stomata in that field of view, here we are getting much less. In fact, if you may count together, the same slide, down a moja, I change the color here, you can see 1, 2, 3. So we have moved from 15 to only 3 stomata. That is how, that's how it can have implications. The magnification that you use, it can have that serious implication. Now, and since an examiner wants you to fill a table, whereby you try once, the second try, and the third try, then you get the average. So what you're supposed to do is to move this slide slightly to left or to right, or you move it up or up and down, so that you can get another field of view. The more you change field of views, the more the answers may vary. The more your counts of stomata may vary. Like now this one, if you move it to another field of view, you may get something like this. Hapa tulikuwa tumepata ngapi? Tulikuwa tumepata 3. Hapa is the same slide, but now with a different field of view, but the same magnification, So here we may now move from only 3 stomata to 1, 2, 3, 4, 5. Hata hii ingine tunaweza sema it is part of us. Although, yeah, 5, 6. You see, the first field of view tukapata 3. The second field of view of the same magnification, we got 6. So that now, how to fill in this table may make sense. Because now, if you look at this table here, the student was supposed now to say field of view 1, 2, 3. At least 3 field of views. Moving the slide, EV kando, EV juu ama kidogo tu. That's how now you land into these 3 readings. The first reading, yeah, the second reading, and the third one. Then you get the average. And that is a manipulation. That's a maneuver that most students hardly know or have an idea. Then after you get the average, now you switch on to the next slide, the upper epidermis, and you do the same. But you must always be conscious of which power, which objective lens does our examiner require us to record from. And maybe that one being very clear, that is an area that we can explore or exploit in future as we prepare ourselves in this kind of questions. Now, maybe at this point, I want to say that in 1997 and also 2016, and the examiner brought another area that has never been given a lot of thought by most of us. And it is good I mention it here. I may forget. We are told to cut. Of course, we were given a mango leaf. It was a mango leaf with a petiole. So we are to make thin section of a petiole. And of course, when you're making sections, we need to be conscious. Is it cross section or transverse section? So sometimes the examiner may be very specific. You want cross section. Like now, this one he wanted us to make cross sections. So you get a petiole of a mango, then you cut like this. Now, remember you are supposed to make as thin sections as possible. Of course, so that we have cells that are not overlapping. Because when cells overlap, light may not pass through and we may not see what we want. Then we are to stain using methylene blue and mount on a microscope slide. But now, after mounting on a microscope slide, and this is now why this question was very unique, we were not observing using a microscope. After doing all that work, we are told to observe using a hard lens. So microscope iliwekwa kando. We made a slide just like the microscope slide, but we used the hand lens to observe. And this is the reason. If it is a stem, this was now a stem, we are likely to observe tissues. Okay, tissues whose arrange the pattern. Here, it may not narrow down to the cell per se. Here, the examiner wanted us to see the pattern of tissues. So this one had nothing to do with cells because you cannot see a cell with your eyes or even with a hard lens. Most hard lenses magnify up to times 10. And by the way, it was possible, even right now it is possible to see the patterns across a petiole of a leaf where we could see the cortex, we could see the epidermis layer, we could see the cortex, and we could see the vascular bundles. Those are tissues, they are not cells. And that pattern is what the examiner wanted. No wonder it was a hand lens. And I hope now you're getting me. So that was KCSE 1997 and 2016. And indeed, what I've enclosed here, this is what was being observed. It was very easy to see this pattern of arrangement of vascular bundles, the cortex, and the epidermis. A monocot now. Of course, the questions that followed was classification to justify which class and the like. So that is an area that also we need to know. Sometimes even a hard lens can give us some results. Now at this point, just close home. In the year 2018, the question of 2018, the practical, that is just how many years ago? 2 years ago or so. It inspired me to think about this apparatus. We call it Lancet. Sijui ni wangapi wanajua Lancet ni nini? Anytime you go to hospital and the doctor wants to take a blood sample, of course he uses a sterile Lancet to prick your finger and a dunga. But it's one of the apparatus that we need to be aware of because we normally use it to make slides of what we call the blood smear. So that's a Lancet, and we use it when sterile, and we use it to prick the finger. And once you prick the finger, you're supposed to press, as I've illustrated there. Sio kutoa damu mingi, kidogo tu, and then you make a blood smear. Now, of course, in 2000 and in KCSE of 2018, it opened a Pandora box of this kind of dimension of questions. By the way, that is what doctors use, or what I call pathologists use when they are making a diagnostic test on malaria. And then they make a blood smear slide. This is a blood smear slide, yeah, which is used to observe the contents inside your blood, whether we can see plasmodium. Yeah. Of course, we are not experts to see it right now, but of course, the trained eye can easily even identify whether it is Plasmodium falciparum or Plasmodium vivax, or whether it's the gametocytes or trophocyte or whatever. But an examiner now can bring a prepared slide. We may not maybe be told to prepare a blood smear, but look at KCSE 2018 question, photograph, paper 3. So below is a photograph of a blood smear from a normal individual. Number one, wale wanafunzi wako wanajua blood smear ni nini. Just to begin with, it put them off. They didn't know now where to turn to. And you see, this is a question of about 15 marks, because every question in paper 3 has around 15 marks out of 40. So most students may have been caught off guard by this one. So it's also good to know these terminologies. As I'm saying, it doesn't harm. In as much as nowadays we may not have a blood smear doing it literally, the examiner will expect us to know what it is and how it is done. So the arrangement is arbitrary, and the number of blood elements is greater than what would normally occur in actual microscope slide. Sasa hiyo ni picha. You know, with now photography, anything is possible. Just like I have shown you the other photograph of a malaria diagnosis. Na hiyo ya malaria diagnosis, uiandike pahali. We need to know even how to do it, even if it's through photographs. Now, we have J, K, and L. And I hope you can see J, K, and L in this blood smear. This is J, this is K, this is L. Wacha niulize mwanafunzi mmoja. By the way, I don't know kama Austin, sijui kama ameingia. There's a young man who had problem with entering the class. Now, Ashley, Jao. Yeah,
Yes.
what do you think J, K, and L are?
J are red blood cells.
Correct?
K is white blood cells.
Exactly.
And L is platelets.
Wonderful. Wonderful. That is Ashley. Thank you so much. So we have red blood cells, that is J. Just from what we know theoretically, how we describe them theoretically, it can easily lead you to remotely know that this is red blood cells, this is white blood cells, or the leukocytes. And you know, one of the reasons is they have lobed nuclei. You can see the dark ring. Their nuclei are not spherical. They are lobed, naive. Then K is platelets, the smallest of the blood cells, the thrombocytes, platelets or thrombocytes. You know, so what I'm saying is, we will talk about types of course, name the parts, and then we state the functions. And you can see now that question had now, so far, those are 3 marks. This is their function, that is 6 marks. So if you don't identify correctly, you'll not get the function. Imagine now losing 6 out of what? 6 out of 40. And of course, the question is very simple. It is based on what you know in theory. Now, types of slides. Now, there are two types of slides. The first type of slide is what we call permanent slides. Then the second one is what we call temporary slides. Mainly what we deal with are temporary slides. But it is upon now the student to know what is the examiner looking for in this question. Now, permanent slides. These are the slides that have been prepared to be used over and over again for a long period of time. The cover strip is usually sealed permanently to enclose the prepared specimen. So most of the permanent slides we are provided with, they are already sealed. And who knows that NEC cannot give us permanent slides, which are already sealed, and then we make observations. So it may not necessarily be photographs, as it was in 2018. NEC can easily supply slides and they can be used in an exam, or we are made to make one because it's not very difficult. On the other hand, we have temporary slides. What we normally observe in our schools are either permanent slides, the ones which are already made, or the ones which are temporary, the ones that we make ourselves. Now there are some terms and techniques that I need to mention. The first one is sectioning. Now, sectioning is the first encounter that is the first thing that you are faced with at the beginning. So in order for a specimen to be seen under a microscope, number one, remember, light has to pass through it. That's why it is necessary for us to obtain very thin, the word is very thin slices or sections of specimen. Now, as we have seen earlier, you can either be told to cut, that is 1997, year 1997, it was cutting, the one for 2016, 1997, we were cutting. Sometimes we are peeling, we peel. If you remember what we saw in 1996, we are peeling the epidermis. So an examiner can tell you to cut. Sometimes he tells you to peel. Although it depends. In 2014, we never cut, we never peeled. We will see what we did. So these sections are made either using a razor blade or what we call a scalpel. Of course, in advanced labs, we use another instrument called microtome. And tutaiona hapo because it's good to have that backdrop. Now, but our blade must be sharp. So here we have got two techniques: very thin sections, so that we allow light to pass through, a sharp blade or a sharp scalpel. This is to avoid distortion. We always know those techniques. The reason why the razor must be sharp is to avoid distorting the cells. Ni kama mtu akikwambia ukate mkate na rula badala ya kisu. You can use a ruler to make slices, but those slices of bread, they will be very distorted. So, no, the reason why you use a sharp razor is to avoid distorting the cells. Why it must be thin to allow light to pass through. That's okay. Then the microtome thing. In advanced lab, we use a microtome. But now, let's go to the next level. Now, sections, of course, if it is a tissue, it can either be longitudinal section or cross section, like the one that we have seen of 2016. It was a cross section. Now, here are the tools. This is now our blade or a scalpel, and sometimes a scalpel may be mounted on a handle. Now, this is our microtome. That is our tool that we call microtome. And since it is mentioned in our Form 1 book, it is good just to know it. So when cutting, we should wet the razor with water. Then we hold the blade horizontally and we move it toward either whatever we are cutting in a way that we produce something that is very thin, as thin as possible. So the more sections we make, the better. So that from there we choose If you just cut once, you'll not provide yourself with the opportunity to choose. It's good to have an opportunity to choose the thinnest among many. And that's a technique that most students always avoid. So make as many, then you choose the thinnest. And even when peeling, there is a way to peel. Peel as many, then you choose among the thinnest. The one that look like indeed it is allowing light to pass through, especially when it comes to epidermis of a leaf. That one you're supposed to slice using very little energy, very little effort, because your blade is sharp. The less effort you use, the higher the chance of getting a very thin section. And that's a technique that I want to repeat. The less effort you use, the more the chance of making a very thin section. Then the one that look like doesn't have anything, any layer of cells, so many layers of cells is the one that you choose. Then the fixation. This is a term. It is well mentioned in our books, especially from one book. And sometimes we ignore. Sometimes we ignore some of these techniques until when it is too late, until when we have been hit. Now, fixation involves treating the specimen with 70% ethanol or a solution of 99% ethanol. That's the first reagent. The second reagent is 1% glacial ethanoic acid. Now, this is the fixative. This is what we call the fixative. The glacial ethanoic acid. This is the fixative. We are fixing so that the structures that we want to observe are maintained. The moment we expose the cells to the vagaries of nature, of environment, they can be distorted. It cannot look the way it should in situ and disturbed. So to avoid disturbing, that's why we are fixing, so that the features that we always know them in theory, we can actually encounter them. So to maintain the structure of the specimen. Number two, to make the specimen to make the specimen material hard enough. Sometimes to make the material hard enough for thin sections to be cut. Now, and I know that this is something that most students don't know. And it's something that we need to take into account. Now, where need be, we may encounter this again, but what we normally know is staining. Now, in most cases, most of the biological specimens that we are handling are transparent. Yeah, it's not something, when something is transparent, when a tissue is transparent, when a cell is transparent, or do I say translucent sometimes, we may not see or we may not identify the parts. So most biological sections are transparent. They need to be stained to make it easier to identify different parts of the specimen. Indeed, we stain to make part of the specimen distinct, as I've indicated there, or to make part of the specimen clear. Like now, nucleus may be transparent. So you're observing and light is passing through the nucleus. You may not see, but when you use a dye, a stain, like iodine solution, methylene blue solution, yeah, the neutral red solution, the eosine, those are some of the common stains that we use. And of course, there are many others. But in high school, we use those three mainly. Now, and it's something that we need to be aware that iodine stain mainly are used when we need those parts of the specimen that can take the stain, especially the structures that are made from carbohydrate. They are likely to be stained with iodine. No single stain has the ability to produce all the results that we may need. Some stains like methylene blue, the one that we used in 2014, they mainly stain the acidic part of the cell, like the nucleus, Then the eosine will stain the alkaline part of the cell, like the cytoplasm. So, and meaning, and we may not have a situation whereby we stain with a compound dye. So sometimes what we see as a cell under a microscope, it may not be the best view possible, because if we use different stains, we will see different structures. So different stains will stain different parts of microscope. And that's why it is good to be aware of this. It depends with what the examiner wants. But given that this is high school, we may only use only one stain per practical. Now, having said that, the language of staining, now we have 4 types of mounts. When I talk of mounts, I'm talking of types of slides that you can be asked to make. Like now, the 2014 was a very interesting one. It was a very interesting one because it was actually my first experience with a wet mount. Tonight, a wet mount. Anyway, the first type of slide is a dry mount. Now, when you talk about dry mount slide, is when we want to observe a hair, nywele, feather, pollen grains. In kitu tu, you don't put a lot of emphasis, because you just put the specimen on the slide and then you observe. You just place a cover slip. But in most cases, we normally experience now the wet mount. The 2014 one was a very interesting one. It was a wet mount, mainly when you're dealing with aquatic samples. In 2014, we just picked our sample from the broth that was respiring. We had a mixture of glucose and water and yeast, yeah, and it had actually been fermenting for some 15 or 20 minutes. Then we had to pick our sample with a dropper. The reason why this one is unique, we have just come from sectioning, where I've said we use a blade, we use a scalpel. Yeah, here we were not cutting anything, yet we made a slide. We used a dropper or a pipette, for that matter, and that's why wet mounts are very interesting. You just pipette, or you drop. You use a dropper and you drop whatever you want to observe on a slide. But since wet mounts are tricky, because the specimen could still be in motion, maybe you're observing plasmodium, or you're observing what? You're observing your cleaner, and it is moving. Yeah? And sometimes you can see it moving literally in the microscope. So you are supposed to observe with the shortest time possible. Like now, the 2014, na tutaiona, most of the cells after some time, they were being immobilized, they were dying. The yeast cells were dying after being exposed to the atmosphere. So most students got frustrated. So you're supposed to take wet mounts very cautiously, because they are moving. Some of the living organisms are dying because they are living. No, they are living, and a living organism can die in a matter of minutes. So you may get a negative result because of how prompt are you to make it and also to observe. Then we have the smear. Now for smear, like now the blood smear, yeah, hiyo tumeona ya 2018. Of course, smears are created by 2 slides. Yeah? Here you have a slide. Maybe you have a drop of blood or whatever you are smearing. Before you place the cover slip, we normally spread whatever we want to observe with a second slide. Then we place the cover slip. So it can be a bit tricky, even as how we place the cover slip. We shall mention, so that we don't trap air bubbles. That one I'll mention down there. Of course, students have been we have seen students making slides, and what they trap are air bubbles. Instead of them observing the cell that we intend them to see, unakuta mwanafunzi amechora air bubble, amekuchorea vizuri na anakwambia hiyo ni bakteria na ni air bubble, hata anakuonyesha mwalimu, ona ile, hata anakuonyesha, ukiangalia unakuta ni air bubble, na hiyo ndio anasema ni cell. So that is something that we need to watch as we move on. Then squash slides. Now, in 1995, this was the most interesting of them all. When you talk of squash slides, of course, squash slides, we make them like in 1995, when we want to actually observe a very, do I say delicate, a very delicate part of a specimen that we want to see, like pollen grains, with the hairs. And we don't want to destroy those pollen grains. We want to see them as they appear in situ. We will see how the 1995 squash slide was made. Even that one for mitosis, for cells that are dividing, it is still a squash slide. Yeah, in fact, it's not 1995, it is 1998, as I have indicated there. It was phenomenal. It was quite tricky. We can read this together so that we understand what is this squash slide. So this is designed for soft samples, very delicate samples. Now, squash slides begins by preparing a wet mount. A wet mount. That is, you are placing the tissue that you want to observe, maybe on top of water, a water drop, yeah, of course, so that the cells don't get dehydrated. We place the tissue over the cover slip. Although now we have already put the drop of water and also stain. Then we place the cover slip. Is the one that they are calling the cover glass. Once we place the cover slip, you don't stop there. You press gently downward, carefully not to destroy the sample or break the cover slip, and squish the sample, remove excess water. So we are pressing the cover slip gently. It can easily break. And as we do so, we squish. We want it to be in a position whereby we are pressing, but we are not destroying. Because if you press hard, like now the pollen grains that we wanted to observe in 1998, they would easily break and we would not observe them. And when they say gentle, it must be gentle. Now, that is, it was quite unique. And by the way, it's very easy to do this. Just getting any flour, and we rub and we dust the anthers as these instructions give. And I think when we are dealing with the practical on flowers, I think I demonstrated this briefly. So remove the anthers, the stamen from specimen M, cut anthers transversely into equal parts. Indeed, this was a Solanum incarnum flower. Tap the pollen grain from the lower part, from the lower half onto the microscope slide. So we are cutting a flower. That is the anther. The anther of Solanum incarnum are very big. The Sodom apple, they are very big. So they can be cut. And then we dust them, or we just tap. Kugonga, pollen grains zinaanguka on the slide. Then we add a drop of iodine solution, which is the stain. Then place the cover slip and press gently to spread the pollen grains, then observe. Now, when you do that kind of a technique, that is what we call a squash slide. Then we are supposed to draw the pollen grains. And it's something that I've done over and over again, and it is a technique that is very simple. And you see now, those are the pollen grains that you are likely to see. You see, when you press so hard, those hooks, those spines are going to break, and you may not see them. And of course, after you observe, maybe you're told to draw. And now from there, you can now predict the agent of pollination because of the hooks or the spines, that is insect pollinated flower. And many other questions now can come from such kind of observation. Now in 2014, this was the broth that students were making, where we had sugar or glucose, and then we were adding yeast. And after we add the yeast, we incubate in a warm water for some time. Of course, we collected the gas, which turned lime water white. But now we were not to use a dropper. This is now the technique that I was saying when we are sometimes dealing with some slides, we obtain a sample without cutting. So put a drop of the content in the boiling tube on a microscope slide. Then we stain with a drop of methylene blue. Then we cover with a cover slip. Then we are supposed to observe under light microscope using low, medium, and high power. And you can see here, the instructions were very clear. In a serial manner, the examiner wants you to move from low to medium to high. Those students who never obeyed the protocol, they got frustrated. Then, of course, you're supposed not to observe and draw what you see. And the high power. So the examiner was in fact interested with high power. Why high power? That's where there was high resolution. The resolution is higher. And you see now, without exploring what has been tested, you may never know that some of these techniques exist. And once you know that there are several types of slide you can make, you can now be keen on what to observe. And you can see, since this one, we have done it so many times, there's one trick that you need to know. Remember, this yeast cell have been respiring for some minutes, about 30 minutes. And when you're told to draw, don't just draw any cell, because some of these cells are already dead. Kama hii unaona ina text yote hii imekufa. But if you see a cell like this one, where my spotlight is, you can see this is a cell which look like what we know it in theory. This is a dividing cell. This is a budding cell. You can see they have not detached. In fact, they are at the last stage of cytokinesis. If you are to deal with mitosis, this is cytokinesis. And you can see even this one has just begun to bud. These are budding cells. Indeed, what we were looking for, students were only expected to draw the cells that are budding. Without that, no mark. And maybe there is something else that I want to say. Anytime that now you're told to draw a cell under a microscope, like now this case, the only way that your drawing can only be valid is when you take into consideration the following. What I'm about to show you here. State the magnification of your drawing. If you draw something that has no magnification, no mark. But there is something that I want us to understand when it comes to magnification. I remember I had told you earlier that the eyepiece lens is interchangeable. And since the eyepiece lens is interchangeable, likewise, the objective lens can also be interchanged by revolving the nose piece. So when you are drawing, or anytime you are dealing with a light microscope, we know that total magnification is a product of eyepiece lens magnification times objective lens magnification. The only valid magnification that the examiner wants is a product of all these. Do we call them, are they combinations or, these possible combinations? Anytime you have a times 10 eyepiece lens, it can only produce, it can actually match with times 40 to give times 400, or times 10, this one, to give times 100, or times 4 to give times 40. Yeah, so these are varied magnifications. Or another possibility, it can also be times 15, whereby if it matches with 4, it gives us, or if it combines with times 4, this one, it gives us times 60. If it is with times 10, it gives us times 150. If it is with times 40, it gives us times 600. So when marking in the magnification that is outside, now when we want high power, the only high power that is valid here is times 400 times 600. But even those ones who use times 150, it is still high enough. But anyway, anything outside this 6 possible total magnification is wrong. It's wrong because it is either this, which is times 10, or times 15 against these 3. So it is good to be aware of such so that you don't come up with a very wild magnification which is out of context. And it's always good to state what magnification is. And here the magnification that we talk about is not the drawing, the one that you have drawn. You know, after you have drawn the yeast, kuna kitu inaitwa drawing magnification. Here we insist on the total magnification from a microscope. If you use the microscope, even after you have drawn, you're supposed to give us the magnification of what you have used. But in case your microscope had any other combination, because it is possible, we can accept it. And of course, that was a yeast cell. Now, during mounting, and I have always said, hey, our time is never enough. Our time is never enough. We have 18 minutes. But it's good to make shorter strides, but we are sure, rather than rush against time. Hatukimbizwi na mtu. Wacha tu tuende pole pole na tuelewe. Now, when we are dealing with mounting, this is another technique that I need to highlight. Now, when you are dealing with mounting, as I've just indicated here, we place whatever we need to place on the slide, and it's always good to do it on a flat bench, because when you mount your specimen on a slanting surface, of course your specimen will spill over. It can actually flow out of the slide. We do it on a flat bench. Hata kama utashika na mkono, it can be a bit dicey. Okay, fine, you can, but we make sure that it is flat, yeah? So horizontal. Then the technique of placing the cover slip is what now most students don't understand. Of course, you are supposed to hold the slide with your hand, and then look at what I'm doing. These 2 fingers are free. These 2 fingers are free. And I don't know why I forgot the slides. Naziko tu pale, but then you place the cover slip. Then using your inoculating needle at an angle of 45 degrees, you now hold the cover slip with the inoculating needle, and then you release it slowly. You slide it in a way, the way I've illustrated here. And the reason why we place the cover slip at an angle, especially 45 degrees, is to make sure that the moment you lower it, as you remove the inoculating needle, you don't trap air bubbles beneath the cover slip, because trapping the air bubbles may actually impair, it will impair your visual ability even to see the specimen that you intend. So this is basically to take care of trapping the air bubbles beneath. It's something that we can purpose to work on. Even here, I can demonstrate. I think I'll do that in our next meeting. And as we do so, we know the role of cover slip. Yeah, the cover slip that we use, it has 3 roles. I know that is in theory, but we can quickly talk about cover slip. Number one, it is to hold the specimen in place. Number two, it is to protect the specimen from dehydration, or even to protect it from dust particles from landing there. And finally, to protect the objective lens. Reason, the specimen could have stain, it will have some water, and it's also having impurities, which in case for some reason the objective lens happens to touch inadvertently, yeah, takama utaki, the objective lens can be stained. And once you are dealing with a stained objective lens, you will see nothing. You will see nothing. Now, I think that one is okay. Now, preparation of temporary slides of a plant cell. Of course, we have talked about peeling. We have talked about spreading the specimen and using a mounting needle on the slide. Then we add water. Of course, we add water so that the specimen is not dry. It's not dehydrated. If it gets dehydrated, it may indeed distort the features that we want to see. Now, having done all that, placing the cover slip, make sure there are no air bubbles. You then observe under low power objective, then you draw the cell. Now, sometimes the examiner will go an extra mile. All that we have said, and may ask you to estimate the cell size. Estimating the cell size. But this is also very tricky. Now, number one, when you are told to estimate the cell size, the first thing that you do is to remember that the units that we use are microns or micrometers. And by micrometers, 1 millimeter is equivalent to 1,000 micrometers. Because in most instances, we will start from the millimeters. Now, the examiner may be interested with the diameter of a cell if the cell is circular. Number two, the examiner may be interested with length of a cell if the cell is rectangular, or width. But most cells are either rectangular or somehow hexagonal. They don't have a very definite shape, as we illustrate in a book. So in most cases, it may not be what we see, but what we assume is length, let it be length. Now, the first thing that you need to remember is to place the transparent ruler on a stage of a microscope across the diameter of the field of view, as I have illustrated there. Once you do that, anytime you place that, and remember we are dealing with the lowest power here, or lowest objective of ×4. But we know, depending on how the question is framed, if we switch from one objective to another, from one magnification to another, whatever we see is a millimeter mark, because our millimeter marks will look like what I'm saying. It will be something very vague. Ukiona kitu kama hii, this is now the millimeter mark. Remember that millimeter mark is magnified, let's say 40 times. It's like that ruler of yours now enlarged 40 times. So whatever you see may be something, utaona to those lines. Make sure you focus until you saw those millimeter marks. Once you see the millimeter mark, if it is one, you put it at the center so that you can see whether there is another one on the edge, another one on the edge. Like now, anytime you see 1 millimeter mark, chances are that those are 2 millimeters. I don't know whether you get that contradiction. Anytime you see one dark line, ukiona one dark line, chances are that the millimeters that across the field of view is 2, because we look at what you call the spaces. So you put that millimeter mark at the center so that you can see whether you can see 2 spaces. Because like here you can see there is like another mark that is about to appear on this end, the left side, another mark that's about to appear on the right side. So here we have 2 millimeter marks. So sorry, 1 millimeter mark, but 2 millimeters. Here we have 1, 2, 3, but according to the way we have aligned them, even the spaces are 1, 2, 3. So here you need to be informed that what the examiner wants is a rough idea of how wide is our field of view. Now, once you count those millimeter spaces, the spaces between millimeter marks, now you multiply whatever you find by 1,000. Like now, this one are 3 millimeters, meaning this field of view is 3,000 micrometers. This is 2,000 micrometers. That is simple. I think that's not a big problem. Then the next level is the same magnification, the same field of view that you have measured and recorded. Now we place our temporary slide with either onion tissue on them, and now we count the total number of cells that occupy the diameter of field of view. Now by this, now we mean that you can, this is what I want us to see, how to count total number of cells that occupy diameter of the field of view. And if you count 3 cells, now you know that the total field of view, if it was 3,000 millimeters, you divide by the number of cells. Then you get the diameter or the length or the width. It depends with the how the cells look like and how the question is framed. But in practice, because this one is more theoretical, I want to see how things may look like in osmosis. Now, under normal circumstances, when you are counting the number of cells across the diameter of field of view, this is what our books always tell us, that you count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Like here we have 12 cells across the diameter of field of view. That is how, in theory, things may look like. But in real sense, this is what you may encounter. This is what you may encounter. No single layer of cells across. It is due not to have an imagination. You have an imaginary diameter. What you need to do is you have an imaginary diameter like this. Of course, my field of view is not circular. Under normal circumstances, the field of view will be circular. Let me try to mute that person. Now, our field of view, when it is circular, we assume that's our diameter, or sorry, I think I should go to the one which is circular. But before I go to the one which is circular, you can see what is happening. When you have low power, you'll see more cells. Yeah. When you have high power, you see fewer cells. So when you're estimating the size, it's also good to do. Yeah. Sorry, it's the network, but it's back. I think we can move on. We are about to close. So I was saying, you realize that low power will give you more cells, high power will give you fewer cells. But the question is, will the field of view affect the diameter of the cell? Will the field of view change the answer? Let's say you used low power and you have so many cells across the diameter. Or you have a high power and you have fewer cells. So what I'm saying is, the higher the power, the fewer the cells. The lower the power, the more the cells, but it will not have an effect. That's the point that I want to mention and remind you. It will not affect the diameter of the field of view. Either way, we'll get the same answer. So that is something that you can think. That is food for thought. So in case the diameter is 1500, that is 1500 millimeters. No, that should be micrometers. That should be micrometers. Then the number of cells are 20. Getting the width, you divide by now the diameter, you divide by the number of cells. In case the length, in case you are looking for the length, because like now in 2001, if you look at the question of 2001, the student were either to estimate the diameter of the field of view, or use the width or use the length. The marking scheme was open. Those who worked with diameter, they could still score. Those who used the width, they could still score. Those who use length, they could still score. But the best approach is to look at the cell and determine which is the best dimension can you get. Come on, width, you use width. Come on, length, you give length. And you state in your answer that mimi ninatafuta width, mimi ninatafuta length. So that now, since answers will vary, what walitumia width, they were marked differently. Those who used length, their answer was marked differently. But those who never mentioned what they are working, who would know, who would tell which dimension they are using? So here we are using what I call the diameter of the field of view divided by the number of cells. The diameter that you get in microns or in micrometers, divide with the number of cells that you see. And the unit must be mentioned accurately. Now, I want to mention the last thing in the interest of time. And it is okay. It's okay to have a lot of information rather than information iishe na kuna muda. Now, in 2001, there is something that students were told to do, and that's what I want us to see. Now, in 2001, so after 2 minutes or 3 minutes to tamaliza, please, if you're the one who has not muted, make sure you mute. Katie. Mute. Now, in 2001, and this is now my last engagement, students were supposed to determine whether osmosis has taken place or not. Now, this is what was the deal. Students were provided with a portion of onion bulbs. They were to remove one leaf from the onion, the one which is more fleshy. So they were to peel the scale leaf. You know, onions have scale leaves, those dry leaves. You cannot obtain a sample from a dry leaf. You're supposed to use the inner tissue, the one which is flesh. You are supposed to peel they were supposed to peel the epidermis of those leaves of an onion, yeah? Ile kitunguu ya round, see ile kitunguu ya majani. Then after they peel the thinnest section, then they are to place a drop of iodine on the edge of the cover slip. Then using a blotting paper, they drain excess iodine solution. Then after they have made that slide and the cover slip is already in place, they were to add water on the edge of the cover slip. You know, the slide is already made. They add water on the edge. And they were to repeat the same now with salt solution. So that now we have two slides. Either it can have water, that is hypotonic solution, ama inaweza kuwa na chumvi. Ukiweka maji ya chumvi, of course, you know what will happen. We are creating a hypotonic solution around the edge of a cover slip. Meaning, in terms of osmosis, when you use salt solution, of course, what will happen is the cells will lose water by osmosis. That's what the examiner wanted them to see in 2001. A very tricky practical. And of course, from there now they were to draw what they see under the microscope, whether it is plasmolysis or whether it is hemolysis. Now, of course, this is the idea. If it is plasmolysis, a plasmolysed cell under microscope, the cell membrane would peel, and they were supposed to draw and indicate this as I have drawn there. You can see outside here, this is the cell wall. To those who are going for other lessons, you are excused. Then in 2-3 minutes, I'll be done. Then you can see here, this is the cell membrane that is peeling off. And of course, the nucleus. You may not see other details in this one. And of course, labeling appropriately. The cytoplasm, the cell wall, the cell membrane, and the nucleus. That's the must you can see. In such a situation, you cannot see anything more. But the point is, under a microscope, as I think I've stopped sharing, under a microscope, this is how you are supposed to observe. And after this, maybe we can call the meeting off, so that those who have left for other meetings, they can do it without feeling they have lost a lot. Under a microscope, this is how it is to show plasmolysis, then what you have drawn. This is a normal cell before it has plasmolized. This is now a plasmolized cell. Mainly you will see from the stain. The stain can show you, because now we only use a stain that will stain the cytoplasm mainly, so that you can see how the cell membrane has peeled off together with the cytoplasm, so that now you can see indeed this is a plasmolized cell. So that what we have produced as a drawing is a justification that indeed plasmolysis has taken place. And that's why it's also good to know a normal cell, how a normal cell would look like, and then how it will now get plasmolized. So 2001 practical, it was another level. And you see, NEC will never shy from bringing a question, just because it is quite, the skills, the manipulation skills are difficult. This was honestly a difficult area. And this is something that we will carry along. Hatutasema hii ndio mwisho, that now that I've talked about it, this is the end. When opportunity arises, I would encourage you to even do this where possible, after we go to school, so that we can see whether it is possible to observe these things. They may look so theoretical, but they're very simple maneuvers. So after the student observed what I can see here, and with the help of the stain, then now they were supposed to draw what I've shown you there. So I think we will stop there, and where need be, we can always apply some of the things that we are saying. So maybe until we meet in our next meeting, we can stop there, and thank you for coming. In case there is an issue, Austin, your hand is up.
Sure, I wanted to ask, when
Yeah.
a student is doing the specimen, what you're seeing in the microscope, do you shade it?
No. Drawing. No. No, we don't shade drawings. In fact, in one of these conferences, we are going to talk about how to draw in biology diagrams. We don't shade. Even if whatever you're drawing is colorful, you don't shade. You just draw what you call outlines. You just make outlines. Yeah. By the way, this one for plasmolysis, this is a very simple experiment, and I would insist where need be, we will talk about it more. By the way, on all those difficult maneuvers to rudia, because repetition is the mother of learning. We may be constrained of time, but where need be, we shall repeat for emphasis. Yeah. So I think we can stop there, and we meet in our next meeting. And there is still a lot of cell physiology we have not talked about, and also microscopy. Maybe in our next meeting, one of these meetings, tutafanya hiyo mixture, areas that we may have missed out the details because of time. Thank you for attending, and be on the lookout of what is coming in our next Saturday. Thank you so much, and God bless you.